Good evening, everyone. I hope I'm audible. ಕೇಳಿಸ್ತಾ ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. Yes. So today's topic is biotechnology principles and processes. Okay. I hope all are doing well. So ಎಲ್ರೂ ಕ್ಲಾಸ್ ಕೇಳಕ್ಕೆ ರೆಡಿ ಇದ್ದೀರಾ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. One of the important chapter so for your board exam as well as KCET. The first thing is, what is biotechnology? So this is a new concept for you people that you didn't, I think. So ಇದನ್ನ ನೀವು ಬೇರೆ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಿಲ್ಲ ಅಂತ ಅನ್ಸುತ್ತೆ. So the word meaning, what is biotechnology? Bio and technology, two words you can find here, ಅಲ್ವಾ? ಸೊ ಇಲ್ಲಿ ಬಯೋ ಅಂಡ್ ಟೆಕ್ನಾಲಜಿ ಟೂ ವರ್ಡ್ಸ್ ಅನ್ನ ನೀವು ನೋಡ್ಬೋದು. ಸೊ ದಿಸ್ ಬಯೋಟೆಕ್ನಾಲಜಿ ವರ್ಡ್ ದಟ್ ಈಸ್ derived from biology and the technology. You know that it is a field in which biological agents and their components have been extracted and used for the generation of useful products. So illi biotechnology. So why this technology is used? So here the living organisms or biological agents and their components and their products, so that have been extracted and used for the generation of useful products. So biology matte technology, so both are involved to produce the useful products to the people. It is a vast technique which has improved and enlarged the scope of modifying organisms and their products for the human and the animals. So ಈ ಒಂದು ಟೆಕ್ನಾಲಜಿಯಲ್ಲಿ that is the vast technology. So ಅಲ್ಲಿ organisms ಅನ್ನ modify ಮಾಡಿ, so that is modified for what purpose? So that is modified for the useful, to obtain the useful products. Okay, so that is called as the biotechnology. Okay, so here further many processes like IVF technique, that is in vitro fertilization and embryo transfer, this IVF technique, and also vaccine preparation process, and also some correction of the defective gene. So ಕೆಲವೊಂದ್ಸಲ ಏನಾಗಿರುತ್ತೆ, ಬರ್ತ್ ಆಗ್ಬೇಕಾದ್ರೆ ಕೆಲವೊಂದು ಡಿಫೆಕ್ಟಿವ್ ಜೀನ್ಸ್ ಇರುತ್ತೆ ಬಾಡಿಯಲ್ಲಿ. ಆ ಡಿಫೆಕ್ಟಿವ್ ಜೀನ್ಸ್ ಅನ್ನ ರಿಮೂವ್ ಮಾಡೋದಿರ್ಬೋದು, ಅಂಡ್ ಇಂಟ್ರೊಡಕ್ಷನ್ ಆಫ್ ದಿ ನ್ಯೂ ಜೀನ್, desired gene to the organisms to obtain the product. So these all processes that comes under the biotechnology. Okay, I hope you got the meaning of the biotechnology meaning ಅರ್ಥ ಆಗಿದೆ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. Mainly, so what are the principles of biotechnology? So in this chapter we are studying principles of biotechnology, principles and processes. Principles means what is the principle of this biotechnology and what are the steps involved in this biotechnology that we are studying. Okay, so the first principle is genetic engineering and second one is the bioprocessing. What is this genetic engineering? I hope you heard this word, genetic engineering. So in this, the techniques that helps to change the DNA or RNA of an organism, so the host becomes phenotypically modified. So genetic engineering alli ಏನು ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ, the gene of the DNA genetic material ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, DNA ಅಥವಾ RNA ಅನ್ನ ಮಾಡಿಫೈ ಮಾಡಿ, ಸೊ ಆ ಒಂದು ಆರ್ಗಾನಿಸಮ್ ಅನ್ನ ಕಂಪ್ಲೀಟ್ ಆಗಿ ಮಾಡಿಫೈ ಮಾಡೋದಕ್ಕೆ that involves the genetic, that comes under the genetic engineering. Okay, then what is bioprocessing? So sterile conditions are maintained to ensure the growth of only desired microbes. It ensures no contamination by any other microbes, thus protecting the product. So bioprocessing, so that also comes under the biotechnology. So here ಕೆಲವೊಂದು ಮೈಕ್ರೋಬ್ಸ್ ಅನ್ನ ಮಾಡಿಫೈಡ್ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಅನ್ನ ಗ್ರೋ ಮಾಡಬೇಕಾದ್ರೆ, ಸೊ ದರ್ ಶುಡ್ ಬಿ ಅ ಸ್ಟೆರೈಲ್ ಕಂಡೀಷನ್. ಸೊ ಸ್ಟೆರೈಲ್ ಕಂಡೀಷನ್ ಅಂದ್ರೆ ಬೇರೆ ಡಿಸೈರ್ಡ್ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ ಬಿಟ್ಟು ಬೇರೆ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಅಲ್ಲಿ ಇರಬಾರ್ದು. ಸೊ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ಬಯೋ ಪ್ರೊಸೆಸಿಂಗ್. ಇಟ್ ಎನ್ಶೋರ್ ದ ನೋ ಕಂಟಾಮಿನೇಷನ್ ಬೈ ಎನಿ ಅದರ್ ಮೈಕ್ರೋಬ್ಸ್, thus protecting the product, so we can get the desirable product. So these are the two principles of the biotechnology. Okay. Next, basic principles of biotechnology. So, basic, why this biotechnology is used. So, you know that the sexual reproduction that forms the variation, and asexual reproduction ಅಲ್ಲಿ ಏನಾಗತ್ತೆ, ವೇರಿಯೇಷನ್ ಆಗೋದಿಲ್ಲ. ಸೊ ವೇರಿಯೇಷನ್ ಆಗೋದಿಲ್ಲ ಅಂದ್ರೆ ಏನರ್ಥ? ಸೊ ದ ಸೇಮ್ ಎಕ್ಸಾಕ್ಟ್ ಸಿಮಿಲರ್ ಕಾಪಿ ಆಫ್ ದಿ ಪೇರೆಂಟಲ್ one is formed as a daughter organisms, right? But in case of the sexual reproduction, you can see the fusion of the gametes and that leads to the variation. So ಇಲ್ಲಿ gametes ಎರಡು ಬೇರೆ ಬೇರೆ gametes fuse ಆಗಿ ಹೊಸ organism form ಆಗೋದ್ರಿಂದ ಏನಾಗುತ್ತೆ, there is changes. Some ಎಕ್ಸಾಕ್ಟ್ ಸಿಮಿಲರ್ ಕಾಪಿ ಆಗಿರೋದಿಲ್ಲ. ಒಂದು ನ್ಯೂ ಆರ್ಗ್ಯಾನಿಸಮ್ ಏನ್ ಫಾರ್ಮೇಶನ್ ಆಗತ್ತೆ ಅಲ್ವಾ, ಅದು ಪೇರೆಂಟ್ ಅಲ್ಲಿಗೆ ಸಿಮಿಲರ್ ಇರುತ್ತೆ ಬಟ್ ನಾಟ್ ದ ಎಕ್ಸಾಕ್ಟ್ ಕಾಪಿ. ಓಕೆ. ಸೊ ಈ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್, ಸೊ ಈ ರೀತಿಯಾಗಿ ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಸೊ ಇನ್ ಅನಿಮಲ್ಸ್ ಆರ್ ಇನ್ ಕೇಸ್ ಆಫ್ ಪ್ಲಾಂಟ್ಸ್, ಸೊ ದಿಸ್ traditional hybridization that may leads to the some undesired character, so they remained in the plant. ಸೊ ಏನಾಗತ್ತೆ, ಈ ರೀತಿಯಾಗಿ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಟ್ರೆಡಿಷನಲ್ ಮೆಥಡ್ ಅಲ್ಲಿ ಆಗಿ ಏನಾಗತ್ತೆ, ಸಮ್ undesired genes they are accumulated in the plants or animals. Okay, so using the biotechnology, what we can do, so here we can remove the undesired gene and we can insert the desired gene, so in that organism. Okay, so that is called as recombinant DNA. So development of recombinant DNA, so that steps involved in the genetic engineering are recombinant DNA, means to remove the undesired characters or some undesirable genes present in the organism, so that new DNA is introduced inside the organism's body, that is called as recombinant DNA. Cloning of the desired gene. ಸೊ ಈಗ ಒಂದು ಜೀನ್ ಅನ್ನ, ಡಿಸೈರ್ಡ್ ಜೀನ್ ಅಂದ್ರೆ ನಮ್ಗೆ ಬೇಕಾಗಿರುವಂತ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಅನ್ನ ಫಾರ್ಮೇಶನ್ ಮಾಡುವಂತ ಜೀನ್ ಅನ್ನ ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಒಳಗೆ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದೇವೆ. ದಟ್ ಶುಡ್ ಗೆಟ್ multiply. Cloning means that should increase their number and transfer of the clone gene into the suitable host organism. Sorry, so cloning of the desired gene, so in the some vector it will get replicate and ಟ್ರಾನ್ಸ್ಫರ್ ಆಫ್ ದ ಕ್ಲೋನ್ಡ್ ಜೀನ್, ಆ ರೆಪ್ಲಿಕೇಶನ್ ಆಗಿರುವಂತ ಜೀನ್ ಅನ್ನ ಸೊ ಬೇರೆ ಬೇರೆ ಆರ್ಗ್ಯಾನಿಸಮ್ಗೆ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡುವಂತದ್ದು. ಓಕೆ, ಸೊ ಹಿಯರ್ ದ ಫಸ್ಟ್ ಸ್ಟೆಪ್ ಇಸ್ recombinant DNA. Recombinant DNA means which contains the desired gene. Okay, so that is introduced, that is multiplied in a ವೆಕ್ಟರ್. ಓಕೆ, ಸೊ ಇಲ್ಲೇನಾಗತ್ತೆ ಅಂದ್ರೆ ಅದು ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಬಾಡಿಯಲ್ಲಿ ಮಲ್ಟಿಪ್ಲೈ ಆಗತ್ತೆ, ಇಟ್ ಇನ್ಕ್ರೀಸ್ ಇಟ್ಸ್ ನಂಬರ್, ದೆನ್ ಇಟ್ ಈಸ್ ಟ್ರಾನ್ಸ್ಫರ್ಡ್ ಟು ದ suitable host organism. Okay, so these all steps are involved in the biotechnology. So in detail we are studying this. Okay, next, so here origin of replication. ಸೊ ಇಲ್ಲಿ ನಾನು ಕ್ಲೋನ್ ಫಾರ್ಮೇಶನ್ ಆಫ್ ದಿ ಕ್ಲೋನ್ಸ್ ಅಥವಾ ಕ್ಲೋನಿಂಗ್ ಆಫ್ ದಿ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಅಂತ ನೋಡಿದ್ವಿ ಅಲ್ವಾ. ಸೊ ಕ್ಲೋನಿಂಗ್ ಆಫ್ ದಿ ಡಿಸೈರ್ಡ್ ಜೀನ್, ಜಸ್ಟ್ ಇಮ್ಯಾಜಿನ್ ದಿಸ್ ಇಸ್ ದ ಡಿಸೈರ್ಡ್ ಜೀನ್. ಓಕೆ, that should be introduced into the host organism. This is the host. ಈ ಒಂದು ಹೋಸ್ಟ್ಗೆ, ಇದೊಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾಕ್ಕೆ ಏನ್ ಮಾಡಬೇಕು, ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಬೇಕು. then this gene that is introduced into the particular organism where it will get multiply. ಸೊ ಮಲ್ಟಿಪ್ಲೈ ಆಗಬೇಕು ಅಂದ್ರೆ ಒಂದು ಇನ್ನೊಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡಿರ್ತಾರೆ, ಅದನ್ನ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರ್ತಾರೆ, ಅಂಡ್ ದಟ್ ವಿಲ್ ಮಿಕ್ಸ್ ವಿತ್ ದ ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ ಆಫ್ ದಿಸ್ ಆರ್ಗ್ಯಾನಿಸಮ್, ಅಂಡ್ ಸೊ ದಟ್ ವಿಲ್ ರೆಪ್ಲಿಕೇಟ್. ಸೊ ಈ ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ ಏನಾಗತ್ತೆ, ಇನ್ನೊಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಬಾಡಿಯಲ್ಲಿ ಜೀನ್ ಜೊತೆ ಮಿಕ್ಸ್ ಆಗಿ ಏನಾಗುತ್ತೆ, ದಟ್ ವಿಲ್ ಅಂಡರ್ಗೋ ರೆಪ್ಲಿಕೇಶನ್. ಸೊ ಇನ್ ರೆಪ್ಲಿಕೇಶನ್ ಯು ನೋ that so origin of replication means where the in which the replication begins, that is called as the origin of replication. Okay, then the foreign DNA that is inserted into the host organism needs to be attached to the origin of relocation, and this results in the formation of multiple copies of the DNA. While if the foreign gene is not attached to the origin of replication, then it may not result in the multiplication of the DNA. So multiplication ಆಗ್ಲಿಕ್ಕೆ ಚಾನ್ಸ್ ಇಲ್ಲ. If origin of replication is not there, then there is no chance of multiplication. Okay, then cloning. Cloning, this is also the process of formation of several identical copies of the DNA template. Cloning, you should know this word. It is the process of formation of the identical copies of the DNA template. Then plasmid. What is this plasmid? An extra chromosomal circular DNA material that helps in the replication of DNA, and they are used in the cloning vectors and also helps in the process of gene expression. See here, a foreign gene is inserted into the plasmid, which then multiplies and results in the formation of several copies of the desired gene. See here, what is plasmid? The thing is, what is plasmid, right? So this is the desired gene. Okay, so I have to get the more copies of this gene to introduce into the host organism. Okay, so I need more copies of this gene. So to get the more copies of this gene, I have introduced in this gene into the bacterial into the bacteria. Okay, just imagine this is the E. coli bacteria. ಈ ಜೀನ್ ಅನ್ನ ನನಗೆ ತುಂಬಾ ಕಾಪೀಸ್ ಬೇಕು, ನನ್ನ ಹತ್ರ ಒಂದೇ ಕಾಪಿ ಇದೆ, ಅದನ್ನ ಮಲ್ಟಿಪ್ಲೈ ಮಾಡಬೇಕು ಅಂತ ಏನ್ ಮಾಡ್ತೇನೆ, ಐ ವಿಲ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಇನ್ಸೈಡ್ ದ bacterial cell. Okay, so this bacterial cell have the circular DNA. This circular DNA that is called as the plasmid. Okay, this circular DNA that is called as the plasmid. So this desired gene that is get mixed with the plasmid DNA. Okay, then it will form the more copies of the desired gene. So now I'll get the, by replication it will form the more copies of the desired gene. Okay, they are used to cloning vectors and also helps in the process of gene expression. Here sometimes this plasmid, ಈ plasmid ಅನ್ನ ಏನಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ as a vector. Okay, so this plasmid which then multiplies and results in the formation of several copies of the desired gene. Okay, so ಒಂದು ಫಾರಿನ್ ಜೀನ್ ಏನಾಗಿರುತ್ತೆ, ಪ್ಲಾಸ್ಮಿಡ್ ಅಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಆಗಿದೆ ಅಂತ ಅನ್ಕೊಂಡ್ರೆ ಇಟ್ ಫಾರ್ಮ್ಸ್ ದ ಸೆವೆರಲ್ ಕಾಪೀಸ್ ಆಫ್ ದಿಸ್ ಡಿಸೈರ್ಡ್ ಜೀನ್. ಓಕೆ, ಸೊ ದಟ್ ಈಸ್ ದ ಪ್ಲಾಸ್ಮಿಡ್. ದೆನ್ ವಾಟ್ ಈಸ್ ಪ್ಲಾಸ್ಮಿಡ್? That is the circular DNA molecule that is present in the bacterial cell and which is also used in the to multiply to get the several copies of the desired gene. So then the desired gene is introduced into the plasmid, then we can get the more copies of the desired gene. Okay, so that is the plasmid. Then antibiotic resistance gene. Okay, So antibiotic resistance. So what is this antibiotic resistance gene? So several microorganisms, for example, bacteria. So this bacteria, they have the gene that resists the antibiotics, means they are able to grow in the some antibiotic media. So they have the ability to resist the antibiotics. Okay. So because of the antibiotic resistance gene. So in certain case of microorganisms, there are several genes that have the ability to grow when there is a specific antibiotic present, while the genes provide resistance against them. So for example, ಇದೊಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಇದಕ್ಕೆ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಕೆಪಾಸಿಟಿ ಇದೆ ಅಂದ್ರೆ, ಸೊ ವಿಚ್ ಹ್ಯಾವ್ ದ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ gene. Okay, so that is called as the antibiotic resistance gene. Then restriction enzymes, very very important. So these restrictions enzymes, they are called as molecular seizures. Okay, see here, molecular seizures. Seizures means, what is the use of the seizure? We use the seizure to cut something, to cut the material, right? Then what is the function of restriction enzymes? They cut the DNA fragments at the specific site. Just imagine, this is the DNA fragment. ಇದೊಂದು DNA fragment ಅಂತ ಅನ್ಕೊಳ್ಳಿ. So it will cut the specific sites of the DNA fragment. So that is the function of the restriction enzyme. These enzymes cut the DNA at the particular site is specific for each restriction enzyme. Okay. Then vectors, already I used this word. Vectors, they are plasmids that help in the process of multiplication and then the transfer of the gene from the organism to the other. So vectors, you know that for the malaria, Anopheles mosquito is the vector. Alva ನಾವು ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ. Vector means what? Which carries the some infecting agents to the human beings. Okay, so in case of the malaria, but here vectors, so the same meaning, which is the carrying some material, something to one organism to the another organism. Okay, so ಇಲ್ಲಿ vectors ಅಂದ್ರೇನು? That is the circular DNA. So that is the plasmid. So we use this vectors to transfer the desired gene to the ಹೋಸ್ಟ್ ಆರ್ಗ್ಯಾನಿಸಮ್. ಓಕೆ, ನಮ್ಮ ಹತ್ರ ಈಗ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಇದೆ, ಆ ಜೀನ್ ಅನ್ನ ಹೋಸ್ಟ್ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಅದನ್ನ ಯೂಸ್ ಮಾಡ್ತೇವೆ. ಅಂಡ್ ಆಲ್ಸೋ ವಿ ಕ್ಯಾನ್ ಗೆಟ್ ದ more copies of the plasmids by this desired genes by this vectors. Okay, so that is the meaning of the vector. So these all are the basic wordings where we use in the recombinant DNA technology and also in biotechnology. Then one more wording, ligase. So you know that the ligase enzyme, that is called as molecular glue, means which um, binds the two fragments of the DNA. That also we studied in the replication alba, replication ಅಲ್ಲಿ study ಮಾಡಿದ್ವಿ. They are the those enzymes that join together the fragment of DNA, that is the fragment. Okay, fragment of DNA that contains the desired gene and the DNA of the host. They help in the sticking of the fragments of the DNA together. So ligase enzyme function ಏನು? That uh, that stick the fragments of the DNA. Okay, then the basic steps in the genetic modification of the organism. Genetic modification andre ಏನು? So if you want to change the gene present in a particular organism, or if you want to introduce particular gene to the organism, so that step, that involves the identification of the desired fragment, introduction of the desired fragment into the suitable host, maintaining foreign DNA in the host, and transfer to the progeny. So, ಇಲ್ಲಿ ನೋಡಿ, as I said, ಸೊ ಈಗ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಾರ್ಮಲ್ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಇದೆ ಜನರೇಷನ್ ಟು ಜನರೇಷನ್. ಓಕೆ, ದೆನ್ ಸಮ್ ಅನ್ಡಿಸೈರೇಬಲ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಸ್ಮಾಲ್, ಈಗ ಪಿ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಆದ್ರೆ ಸ್ಮಾಲ್ ಸೀಡ್ಸ್ ಫಾರ್ಮೇಶನ್ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ದ ಅನ್ಡಿಸೈರ್ಡ್ ಕ್ಯಾರೆಕ್ಟರ್ ದಟ್ ಈಸ್ ಅಕ್ಯುಮುಲೇಟೆಡ್ ಇನ್ ದ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, so to remove this, the genes responsible for the small seeds, so that is removed, and the gene which is responsible for the big seeds, so that is introduced into the plant. Okay, then so from the next generation we will get the more plants with the big size seed. Okay, that is the need of this recombinant or the modification of the gene. Identification of the desired DNA fragment, introduction of the desired DNA fragment into a suitable host, maintaining the foreign DNA in the host, and its transfer to the progeny. Okay, so these all steps we are studying in detail. Okay, so I'm not going to explain these steps. Yes, recognition sequences, the basis of the DNA sequences that are specific for each restriction enzymes and act as a site for the restriction cutting or resulting in the formation of palindromic sequence. So recognition sequences. So particular gene, just imagine this is the DNA. So the particular restriction enzymes cut the gene in the particular site, so that is called as recognition sequences. Okay, and that is particular for particular restriction enzymes. So ಇದನ್ನ ನಾನು next explain ಮಾಡ್ತೇನೆ, ಯಾಕಂದ್ರೆ so in detail we are studying about the recombinant DNA technology. Okay, here, the process of recombinant DNA technology. What is this recombinant DNA technology? Okay, so recombinant andre new combination of the DNA. Okay, just imagine this is the host DNA. Host ಅಲ್ಲಿ ಏನಿದೆ ಈ DNA ಇದೆ. So it has some undesirable character, or we have to introduce some new genes to this. Okay, so we have added the new gene to the basic DNA molecule. So this new gene that is responsible for the some desired character. Okay, so this is very easy introduction of the some desired gene into the host gene. Okay, but this is called as the recombinant DNA technology. So what are the steps involved in this recombinant DNA technology? We'll see. Okay, ನೋಡಿ ಯಾವ steps ಇದೆ ಅಂತ. The first step is DNA with desired characteristic is isolated from the organism. So the desired DNA ನಮಗೆ ಬೇಕಾಗಿರುವಂತ ಕ್ಯಾರೆಕ್ಟರ್ ಇರುವಂತ ಒಂದು ಜೀನ್ ಅನ್ನ, so that we have to isolate. Okay, I have isolated the desired DNA from the organism. Second step is the desired DNA is integrated with the DNA of the vector. This is done, the DNA fragment needs to be replicated. So this gene I have introduced inside the vector. Okay, just imagine this is the gene that is the desired gene. Okay, blue color ಅಲ್ಲಿ ಇದನ್ನ introduce ಮಾಡ್ತಾ ಇದ್ದೇನೆ, blue color. ಸೊ ಈ ಬ್ಲೂ ಕಲರ್ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಅನ್ನ ವೆಕ್ಟರ್ ಅಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದೇವೆ. ಓಕೆ, ಸೊ ಹಿಯರ್ ಇಟ್ ವಿಲ್ ಗೆಟ್ ಮಲ್ಟಿಪ್ಲೈ. ಸೊ ಇಲ್ಲೇನಾಗತ್ತೆ, ಅದರ ನಂಬರ್ ಏನಾಗತ್ತೆ, ಇನ್ಕ್ರೀಸ್ ಆಗತ್ತೆ. ದೆನ್ ದಿಸ್ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಕಂಟೈನಿಂಗ್ ವೆಕ್ಟರ್, ದ ಡಿಸೈರ್ಡ್ DNA is linked with the origin of replication of the vector, and thus it is multiplies itself in the host. So that will get multiply in the host. Okay, then the newly formed DNA is the recombinant DNA. That newly formed DNA, that is called as the recombinant one, and this DNA is inserted into an organism to incorporate with the desired characteristics. So now we have to introduce into the host. Just imagine any animal or plant. So this gene that is incorporated with the host gene and that produces the new characters. So that organisms we can call as genetically modified organisms. Okay, so genetically modified organisms means some extra desirable gene is transferred to the organism to get the desirable product. So they are called as genetically modified organisms, GMO. Next, the tools of recombinant DNA technology. Yes, we'll perform this recombinant DNA, but some tools are required to perform this experiment. Okay, recombinant DNA technology. The first one is restriction enzymes. So restriction enzymes means molecular seizures. These are the enzymes. It's also called as molecular seizures. Here you can see what is the function of this restriction enzymes. These are the enzymes which cut the DNA at the specific sites into fragments. So illy, just imagine this is the gene present in a particular organism, but we need to cut specific sequence. We need this particular region of this gene, this DNA. So that is done by the restriction enzymes. Okay, they belong to the class enzyme called nuclease. Then naming of the restriction enzyme. Okay, so there are many restriction enzymes. EcoRI is one. EcoR1 is the one example for that. Here how this naming of the restriction enzymes will be done. The first letter indicates the genus. So here EcoR1 means this E. coli bacteria. Okay. Second two letters indicate specific of the prokaryotic cell from they were isolated. So from where they were isolated, so the next two cells as spellings that will indicate. For example, eco R1 comes from E. coli RY13. So that eco R1 that is isolated from the E. coli bacteria. So E. coli, Escherichia coli bacteria, right? E. coli bacteria. So we use the species name. Okay. So this is the E means genus name, CO means species name. Then R means in which type of strains. R means strain. What type of the strain they have, that is the R. Okay. And 1. What is this 1? Roman number, the order in which enzymes were isolated from the strain of the bacteria. Okay, so how in which order they isolated from that bacteria, that is the 1. Okay, eco R3. So like that they'll give the number. Okay, again one more, HIND3. Okay, so here H represents the genus name, IN that is the species name, then D that is the state, and 3 is the order of isolation. Okay, so that is the thing. Then so in restriction enzymes there are two types. The first type is exonuclease and second type is endonuclease. So exo means outside, endo means inside. Okay, so exo means what? Outside. You know that they remove the nucleotides from the ends of the DNA to cut the ends of the DNA. Just imagine this is the ends of the DNA. To cut the ends of the DNA, so these exonucleases are used. Okay, endonucleases, they cut the specific position with DNA. So they bind the specific recognition sequence at the DNA and cut the two strands at the specific points. So endonuclease, they cut the specific points inside the DNA. Okay, so that is exonuclease and the endonuclease. That is the two types of the restriction enzymes. Okay, then the first restriction endonuclease is HIND2. Okay, so already I told the example. It cuts the DNA by recognizing a specific sequence of 6 base pairs. So this sequence is called as recognition sequence for the HIND2. Okay, so these enzymes that identify the particular sequences and which cuts the particular sequences of the DNA, that is called as the recognition sites. Okay, so that is a type of endonuclease enzyme. Then, so here restriction enzymes, they cut the nucleotide at the palindromic nucleotide sequence. ಸೊ ಈ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ಏನಿದೆ ಅಲ್ವಾ, ಇದು ಪರ್ಟಿಕ್ಯುಲರ್ ಸೈಟ್ಸ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡುತ್ತೆ, ಡಿ ಎನ್ ಎ ಮಾಲಿಕ್ಯೂಲ್ ಅನ್ನ ಕಟ್ ಮಾಡುತ್ತೆ. ಸೊ ಪರ್ಟಿಕ್ಯುಲರ್ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, ಎಂಡ್ಸ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡಲ್ಲ, ಡಿ ಎನ್ ಎ ಪರ್ಟಿಕ್ಯುಲರ್ ಸೈಟ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡುತ್ತೆ. ದಟ್ ಸೈಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಪ್ಯಾಲಿಂಡ್ರೋಮಿಕ್ ಸೀಕ್ವೆನ್ಸ್. So what is this palindromic sequence? So this is the DNA fragment. So just imagine here the nucleotides are arranged G, A, guanine, adenine, thymine and cytosine. Okay. So again I'll write guanine, adenine, thymine, cytosine. So palindromic sequences means what? Which read from one side forward and the backward same. Okay, so if we read something, so that is forward and the backward, that has the same spellings. For example, T A T A means here also T A T A. So in forward direction also T A T A, in backward direction also T A T A. So that is called as the palindromic sequence. Okay, so here the restriction enzymes, they cut at the palindromic sequence. See here GATTC, and read this backward GAATTC, GAATTC. So this is the palindromic sequence. Okay, so they cut at the region where the palindromic sequences are present. So that is called as the restriction. Okay, then restriction enzymes cut the strand a little away from the center of the palindrome sites, but between the same two bases on the opposite strands. This leaves the single stranded overhanging stretches at the ends, that is called as sticky ends. Just imagine, see here, GAATTC is there. So, and this is the GATTC. So, completely this segment is not removed by the restriction, not cut by the, which do not cut this complete restriction side. It leaves some sticky ends. See here. So, this AATTC is removed, and here, so in again, this part is just only this part is removed and this sticky end is remaining here. CG is remaining there. Okay, so here only AATTC is removed by this. Okay, so this end, so that is called as the sticky end. Okay, so here also it formed the sticky end. So illu ಏನನ್ನ ಕಟ್ ಮಾಡಿದೆ, CATTA. ಇಲ್ಲಿ ನೋಡಿ, ಈ ಒಂದು ಸೀಕ್ವೆನ್ಸ್ ಅಲ್ಲಿ ಏನ್ ಕಟ್ ಮಾಡಿದೆ, ಕೆಳಗಡೆ ಕೆಳಗಡೆ ಟಿ ಟಿ ಎ ಅಲ್ವಾ, ಸೊ ಇಷ್ಟನ್ನ ಕಟ್ ಮಾಡಿದೆ. ದೆನ್ ಸೊ ಇಟ್ ಫಾರ್ಮ್ಸ್ ದ ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್. ಎರಡೂ ಕಡೆ ಏನಾಗತ್ತೆ, ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ ಫಾರ್ಮೇಶನ್ ಆಗತ್ತೆ ಅಲ್ವಾ. ಸೊ you can see the sticky ends here. Then the new one is the new desired DNA binds this region of the DNA molecule. Okay, so complete this segment will be not removed, only it forms the sticky ends. Okay, leaving the half of the nucleotides and single strands, overhanging stretches at the ends. So sticky ends form hydrogen bond with the complementary cut counterparts. This stickiness facilitates action of the ligase. To bind this sticky ends, the DNA ligase is required. Okay. When cut by the same restriction enzymes, the DNA fragments have the same kind of the sticky ends. So you can see the AATT and here also TTAA, means same kind of the sticky ends you can see. So that is joined by the DNA ligase. Okay. So here this is the graphical representation of the recombinant DNA technology. Very important for your exam. Okay, diagram is very important. See here, this is the foreign DNA. Foreign DNA means that is desired DNA. Okay, same restriction enzymes cutting both foreign DNA vector and DNA at the specific point. Okay, so il nodi, so the restriction enzymes that cuts the particular site of the palindromic sequence of the particular gene desired DNA. Okay, so this is the desired fragment that I have to introduce into the vector. So here also the some fragment is removed, so it is also having the sticky ends and it is also having the sticky ends. So these both are joined by the ligase. Okay, now it is introduced inside the, this vector is introduced inside the host. Okay, so that is the transformations. Here you can see the multiplication of the cells. Okay, that is called as the recombinant DNA technology. That is the cloning host, means where the multiplication of the desired genes takes place. ಇಲ್ಲೇನಾಗಿದೆ ನೋಡಿ, ಈ ಒಂದು ವೆಕ್ಟರ್ ಏನ್ ಮಾಡಿದ್ದಾರೆ, ಸಾರಿ ಪ್ಲಾಸ್ಮಿಡ್ ಅನ್ನ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದಾಗ, ಸೊ ದಟ್ ವಿಲ್ ಗೆಟ್ ಮಲ್ಟಿಪ್ಲೈ. ಓಕೆ, ನಂಬರ್ ಏನಾಗಿದೆ, ಇನ್ಕ್ರೀಸ್ ಆಗಿದೆ ಅಲ್ವಾ. ಸೊ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ರಿಕಾಂಬಿನೆಂಟ್ DNA ಟೆಕ್ನಾಲಜಿ. ನೆಕ್ಸ್ಟ್, separation and isolation of the DNA fragments. So how to separate and isolate the DNA fragments while introduce it into the particular vector. Okay, so ಇಲ್ಲಿ ನಮ್ಮ ಹತ್ರ ಏನಿದೆ, gene ಇದೆ. Okay, sorry, DNA ಇದೆ. So DNA molecule that we should cut, okay, by using the restriction enzyme. So the cutting of DNA by restriction endonuclease results in the fragments of the DNA. So it will form the fragments of the DNA. Okay, then these fragments can be separated by technique known as the gel electrophoresis. ನಮ್ಮ ಹತ್ರ ಬೇರೆ ಬೇರೆ size DNA fragments ಇದೆ, but we have to separate that. Okay, we have to separate that to introduce into the host. So since the DNA fragments are negatively charged, so that separation technique is known as gel electrophoresis technique. Okay, what is this gel electrophoresis? Let us see the process. That is one of the very important one. Since DNA fragments are negatively charged, molecules They can be separated by forcing them to move towards the anode under the electric field through the medium or the matrix. Okay. So here we use the gel electrophoresis process. So here we put the DNA fragments into these wells, and you know that DNA is negatively charged molecule. So we can separate them by using the by making them to move towards the positively charged anode. Okay, so this is the gel electrophoresis unit. So here I keep the DNA fragments, and I'll connect this to the electric bulb. Okay, electric battery. So when we pass the current through this, the negatively charged DNA molecules, they move towards the positively charged anode. Okay, but the DNA fragments separate according to their size through the sieving effect provided by the agarose gel. So ಇಲ್ಲಿ ನಾವು ಅಗರೋಸ್ ಜೆಲ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿರ್ತೇವೆ. ಅಗರೋಸ್ ಜೆಲ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿರೋದ್ರಿಂದ ಏನಾಗತ್ತೆ, the smaller the fragment in size, they farther it moves. Small fragments ಏನಾಗತ್ತೆ, ಫಾಸ್ಟ್ ಆಗಿ ಮೂವ್ ಆಗತ್ತೆ. ಲಾರ್ಜರ್ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ they move slowly. Okay, so why this gel electrophoresis matrix is used to separate the DNA fragments. ನಮ್ಮ ಹತ್ರ ಬೇರೆ ಬೇರೆ ಸೈಜ್ DNA fragments ಇದೆ ಅಲ್ವಾ, ಸೊ ಅದನ್ನ ಸೆಪರೇಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಅಗರೋಸ್ ಜೆಲ್ ಅಲ್ಲಿ ನಾವೇನ್ ಮಾಡಿದ್ದೇವೆ, ಈ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಅನ್ನ ಹಾಕಿದ್ದೇವೆ. ವೆನ್ ವಿ ಪಾಸ್ ದ ಎಲೆಕ್ಟ್ರಿಸಿಟಿ, the smaller fragments, they move fastly towards the anode, and the larger fragments, they remain in the anodes one side. Okay, so they move slowly. Then the separated DNA fragments can be visualized. So just imagine this is the agarose gel unit. So here you can see the DNA fragments separated like this. Okay, so visualized only after staining the DNA with compound known as ethidium bromide. Ethidium bromide staining is used. So when this stain is used, and when we expose the DNA under the UV radiation, so you can see the bright orange colored bands here. Orange color bands you can see under the UV radiation, and DNA in the ethidium bromide stain gel exposed to UV light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This is known as illusion. So the desired separated DNA segment, so just imagine this is the desired DNA segment, so that is extracted from this, and that is known as elution process. The DNA fragments purified in this way are used to constructing the recombinant DNA by joining them with the cloning vectors. So here these DNAs that are used to form these DNA fragments are uh, used in the recombinant DNA technology. Okay, so this is how DNA fragments are separated. Okay, then one of the important thing that is used in the recombinant DNA technology, that is the cloning vector. So as the name says, cloning vector. So what is the meaning of cloning vector? Cloning means make the more copies of the DNA, right? It is a DNA molecule that can carry a foreign DNA segment and replicate inside the host cell. Example, plasmids and bacteriophage. So what is this cloning vector? It is the DNA molecule. Okay. So just imagine circular or DNA molecule that can carry the foreign DNA. So that is able to carry the foreign DNA to the inside the host cell. Okay. So plasmids and bacteriophages. So this is the bacterial DNA and this is the bacterial plasmid that is act as a vector, and this is the bacteriophage and bacterial DNA act as a vector. Okay. Sorry, this is the virus, bacteriophage virus, and which act as a uh, replication. Okay, which comes in the replication process. Then plasmids are autonomously replicating extra chromosomal bacteria, DNA of the bacteria. Some plasmids have only 1 to 2 copies per cell and otherwise 15 to 100 copies per cell. So plasmid, that is the circular autonomously replicating, and the extra chromosomal DNA. Okay, so that is the genetic material, the chromosomal material that is present in the bacteria. And bacteriophages so have very high copy number of their genome with the bacterial cells. Then, so the very important, what are the features of this cloning vector? Okay, let us see the important features of this cloning vector. Cloning vector's important features ಏನು? So this cloning vector which has origin of replication or ORI. So ORI means where the replication begins. Okay, so here in this very good example is PBR322 vector. Here you can see the origin of replication. Okay, from where the replication begins. And selectable marker. Okay, so the marker, selectable markers you can see, and the cloning sites. Cloning sites you can see here, this BAMH SAL1 and EcoRI1. So these all are the sites where the restriction enzymes they cut the PBR322. Okay, ಇದೆಲ್ಲ ಏನಿದು? ಸೊ EcoRI1 HIND3 ಅಲ್ವಾ? ಸೊ ಇಲ್ಲಿ ಈ ಒಂದು ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ ಏನಿದೆ ಅಲ್ವಾ, ಸೊ ದಟ್ ಬೈಂಡ್ಸ್ ದಿಸ್ ರೀಜನ್ ಅಂಡ್ ವಿಚ್ ಮೇಕ್ ದ ಕಟ್ಸ್ ಇನ್ ದಿಸ್ ರೀಜನ್. ಓಕೆ, ಇಲ್ಲಿ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ಸ್, ಈ ಒಂದು ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ಸ್ ಈ ಪರ್ಟಿಕ್ಯುಲರ್ ರೀಜನ್ ಅನ್ನ ಕಟ್ ಮಾಡುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ದ ಮೀನಿಂಗ್. And the cloning sites and the new foreign DNA is introduced in this site. Vectors for cloning genes in plants and animals. Okay, so these all are the important structures that you can observe in the PBR 322. Let us see the what is origin of replication. Okay, so it is a sequence where the replication starts. It is a sequence where the replication begins. So you can see the ORI here. Okay. A piece of DNA linked to ORI can replicate within the host cells. It also controls the copy number of linked DNA so that getting many copies of target DNA, it should be cloned in a vector whose origin support high copy number. Okay, so where just remember, this is the origin of replication from where the replication begins to increase its copy number. Then selectable marker. So what is this selectable marker? Marker gene. It is a gene that helps to select the transformants and eliminate the non-transformants. Okay, so just imagine this is the recombinant plasmid. So when we introduce this recombinant plasmid into the host, okay, so there are some genes, so that is helpful to select the transformants and the non-transformants. So here recombinant DNA is present in a particular cell, means that is the transformant cell. To select this transformant cell from the non-transformant cell, so the selectable markers are helping. If a piece of DNA is introduced in a host bacterium, it is called as the transformation. Such bacterium is the transformant. If the transformation does not take place, it is non-transformant. Okay. ಈಗ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಶನ್ ಆಗಲಿಲ್ಲ, ಜಸ್ಟ್ ಇಮ್ಯಾಜಿನ್ ದಿಸ್ ಈ ಕೋಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ದರ್ ಇಸ್ ನೋ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಶನ್. ಓಕೆ, ಸೊ then this bacteria, E. coli bacteria, is called as the non-transformant. Then selectable markers. So selectable markers of E. coli includes the genes, that is antibiotic resistance genes like ampicillin, chloroamphenicol, tetracycline and kanamycin. Okay, normal E. coli cells have no resistance against these antibiotics. So to select the transformant and the non-transformant, so the selectable marker genes are helpful. Okay. Then cloning sites. So what are these cloning sites? To link the alien DNA, the vector needs to a single or very few recognition sites for the restriction enzymes, and more than one recognition sites generate the several fragments. It complicates the gene cloning. So illi recognition sites, so these all are called as recognition sites by the different restriction enzymes. So ಇಲ್ಲಿ ಬೇರೆ ಬೇರೆ restriction enzymes ಏನ್ ಮಾಡುತ್ತೆ, so they cut the particular region and they introduce the new gene in this particular sites, that is called as the recognition sites. Then, or cloning sites, ligation of alien DNA is carried out at a restriction site present in one or two antibiotic resistance gene. So illi the ligation of alien DNA. So ಈಗ ಒಂದು alien DNA or foreign DNA, so that should be introduced into this PBR 322. So for example, ligation of foreign DNA at the BAMH1. Okay, so here this is BAMH1. So site of tetracycline resistance gene in a vector PBR 322. As a result, recombinant plasmid is formed. If a ligation does not occur, it is called as the non-recombinant plasmid. So if you ligate, means using the ligase enzyme, so if you fix the foreign DNA in this site where the BAMH1 site, so if the tetracycline gene is present in this region, so that is removed. Okay, so ಈ ಒಂದು region ಅಲ್ಲಿ BAMH restriction enzymes ಏನ್ ಮಾಡಿದೆ, ಈ ಒಂದು enzyme ಈ ಒಂದು region ಅನ್ನ ಕಟ್ ಮಾಡಿದೆ ಅಲ್ವಾ. ಈ region ಅನ್ನ ಕಟ್ ಮಾಡಿ ಏನ್ ಮಾಡಿದೆ, foreign DNA ನ introduce ಮಾಡಿದೆ ಅಲ್ವಾ, by using the ligase enzyme. So the genes, so sometimes that have the some antibiotic resistance gene. So this antibiotic resistance genes, that is removed. Okay. When a foreign DNA is inserted with a gene of a bacteria, that gene inactivated, it is called as insertional inactivation. Here recombinant plasmids lose tetracycline resistance due to the insertion of the foreign DNA. So when the plasmids are introduced into the E. coli, all three types of cells are formed. Okay. ಇಲ್ಲಿ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಸೈಟ್ ಅನ್ನ ಯಾವುದನ್ನ ನಾವು ಏನ್ ಮಾಡಿದ್ದೇವೆ, ಇಲ್ಲಿ ಕಟ್ ಮಾಡಿದ್ದೇವೆ, ದಟ್ ಇಸ್ ದ BAMH 1. ಸೊ ದಟ್ ಇಸ್ ದ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ಆಗಿತ್ತು ಅಲ್ವಾ, TETR ಅಂತ ಇತ್ತು ಅಲ್ವಾ. ಈ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ಅನ್ನ ನಾವು ತೆಗೆದಿರೋದ್ರಿಂದ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ದಟ್ ಈಸ್ ನಾಟ್ ಏಬಲ್ ಟು ಗ್ರೋ ಇನ್ ದ tetracycline, that is not have the ability to resist the tetracycline. ಆ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಅಲ್ಲಿ ಅದೇನ್ ಮಾಡೋದಿಲ್ಲ, that is not able to grow. Okay, so just imagine this is the PBR 322, okay, which has the origin of replication, where the replication begins, and which has the some genes that is the selectable markers and that are useful to antibiotic resistance genes. For example, tetracycline resistance genes, ampicillin resistance genes, means they are able to grow in the ampicillin and the tetracycline. ಈಗ ಈ ಒಂದು ಜೀನ್ ಅನ್ನ ರಿಮೂವ್ ಮಾಡಿ by some enzyme and new site is new one is introduced here. Okay, ಇಲ್ಲಿ desirable ಒಂದು ಡಿಸೈರೇಬಲ್ ಜೀನ್ ಅನ್ನ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರೋದ್ರಿಂದ ಏನಾಗಿದೆ, ದ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ಇಸ್ ರಿಮೂವ್ಡ್. ಓಕೆ, ಈ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ಏನಾಗಿದೆ, ರಿಮೂವ್ ಆಗಿದೆ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಇನ್ಸರ್ಷನಲ್ ಇನ್ಆಕ್ಟಿವೇಶನ್. Here the recombinant plasmids lose tetracycline resistance due to the insertion of foreign DNA. See here, so this bacteria, that is the transformed bacteria, and here you can see non-transfer bacteria also. So if we are growing this ampicillin media, so in the ampicillin media both the cells, so they'll grow. Okay, so this non-transform cell, they don't have the ampicillin resistance gene, but these have the ampicillin resistance gene. So these bacteria are only able to grow and form the colonies. ನೋಡಿ, ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಗ್ರೋ ಆಗೋದಿಲ್ಲ ಈ ಮೀಡಿಯಾದಲ್ಲಿ. ಯಾಕೆ? ಆಂಪಿಸೆಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಮಾಡಲಿಕ್ಕೆ ರಿಕಾಂಬಿನೆಂಟ್ ಇಲ್ಲ ಅಲ್ವಾ. Then non-transformants, 3 types of the cells, right? They have no plasmid, so they are not resistant to either tetracycline or the ampicillin. Non-transformed bacteria ಏನಾಯ್ತು, they don't have any genes to resist the some transformants with non recombinant plasmid. They are resistant to both tetracycline and the ampicillin. Okay, transformants means, so they have the both resistant to the tetracycline and the ampicillin. Transformants with recombinant plasmid, they have resistant to only ampicillin because tetracycline so gene is removed. Okay, recombinant plasmid ಅಲ್ಲಿ ಏನಿಲ್ಲ, ಆಂಪಿಸಿಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ಇದೆ, ಬಟ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಜೀನ್ ಅನ್ನ ಏನ್ ಮಾಡಿದ್ದಾರೆ, ರಿಮೂವ್ ಮಾಡಿದ್ದಾರೆ ಅಲ್ವಾ. ಸೊ ದೀಸ್ 3 we can see here. So by using this we can select the recombinant plasmids can be selected out from the non-recombinant. So ಇಲ್ಲಿ ನಾವು ಈ ಕ್ಲೋನಿಂಗ್ ವೆಕ್ಟರ್ಸ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು we can see the extract the recombinants and the non-recombinants or transformants or the non-transformants in the medium. Okay, the recombinants grow in ampicillin medium but not in the tetracycline medium, that you know. Thus one antibiotic resistant genes that helps to select the transformants. Okay, so this is about the PBR 322 selectable marker. Okay, so the remaining things I will explain in the next class. So with example I will explain this. Okay, yes, so these are the some features of this cloning vector. The remaining features about this vector I'll explain in the next class. Thank you.